Welcome to G-Fun Facts Online. Let us dive straight into one of the most fiercely protected rules in all of science. For over half a century, the foundational rules of molecular biology were treated with the kind of reverence usually reserved for ancient religious texts. At the heart of this biological doctrine lies the central dogma and the universal genetic code. This code is essentially a microscopic dictionary that translates the language of DNA into the language of proteins, the physical building blocks of life. According to the classic biology textbooks sitting in classrooms around the world, this translation is absolute, precise, and completely unambiguous. In the 1960s, scientists cracked this genetic code, a monumental achievement that revealed exactly how life constructs itself from scratch. They discovered that the genetic alphabet consists of just 4 chemical letters: adenine, cytosine, guanine, and thymine in DNA, with uracil replacing thymine in the messenger RNA. Because the proteins that make up your muscles, enzymes, and tissues are built from 20 standard amino acids, and there are only 4 genetic letters, the cell has to read the genetic script in 3-letter words, which biologists call codons. Mathematically, if you arrange 4 letters into groups of 3, you get exactly 64 possible combinations. 61 of these codons specify a particular amino acid. The remaining 3, known historically as ochre, amber, and opal, or UAA, UAG, and UGA, do not code for any amino acid at all. Instead, they act as the periods at the end of a genetic sentence. They are the universal stop codons. When the cell's protein manufacturing factories, which are massive molecular machines called ribosomes, are chugging along a strand of messenger RNA building a protein, they eventually encounter one of these 3 sequence combinations. When they hit a stop codon, they summon a specialized protein called a release factor. This release factor acts like a pair of molecular scissors. It snips the newly synthesized protein chain free so it can fold into its functional 3-dimensional shape and go do its job in the cell. Under the classic paradigm of biology, a stop codon is absolute. It is a digital command. If a genetic mutation accidentally places a premature stop codon in the middle of a gene, which scientists call a nonsense mutation, the ribosome halts prematurely. The result is a truncated, usually useless, and sometimes highly toxic protein fragment floating around the cell. In humans, such genetic typos are the root cause of devastating diseases like cystic fibrosis and Duchenne muscular dystrophy. Because of the sheer danger of making half-finished proteins, evolutionary pressure relentlessly demands that stop codons be interpreted unambiguously. A stop means stop, perfectly, every single time. Or so we thought. But nature, it turns out, has a profound disdain for absolute rules. Deep within the murky depths of hydrothermal vents, the oxygen-deprived muck of swamps, and even the dark corridors of the human digestive tract, an ancient lineage of microorganisms known as Archaea is quietly rewriting the textbook. To understand this, we have to travel back to 1977, when the biophysicist Carl Woese shocked the scientific community. He revealed that what we previously grouped together as simple bacteria actually consisted of 2 completely distinct domains of life: bacteria and archaea. Archaea look like bacteria under a microscope, but genetically and biochemically, they are as different from bacteria as you and I are. Archaea are famous for their extremophilic lifestyles. They inhabit the most inhospitable environments on Earth. We are talking about boiling acid springs in Yellowstone National Park, hypersaline lakes, and crushing oceanic trenches where sunlight never reaches. To survive these extremes, Archaea have evolved bizarre biochemical adaptations, from cell membranes constructed of unique ether lipids that resist melting, to DNA repair mechanisms that can stitch together a completely shattered genome. But among their most fascinating adaptations is their ability to manipulate the universal genetic code. Some Archaea, specifically methanogens, which are methane-producing microbes, have expanded their genetic alphabet beyond the standard 20 amino acids. They have actually annexed the universal stop codons, hijacking them to encode entirely new, exotic amino acids. The first crack in the universal genetic code appeared with the discovery of selenocysteine, often dubbed the 21st amino acid. Found across all 3 domains of life, but highly prominent in certain archaeal and bacterial species. Selenocysteine is encoded by the UGA, or opal, stop codon. You might be wondering, how does the ribosome know whether UGA means stop and cut the protein, or ignore the stop and insert selenocysteine? The answer lies in a highly sophisticated structural workaround. In organisms that use this 21st amino acid, the messenger RNA contains a specialized hairpin loop structure known as the selenocysteine insertion sequence element. In Archaea, this element is typically located in the untranslated region of the RNA, sometimes hundreds of nucleotides away from the actual UGA stop codon. When the ribosome arrives at the UGA sequence, a specialized elongation factor protein binds to that hairpin loop and physically drags a custom-built selenocysteine transfer RNA into the ribosome. This structural acrobatics forcefully overrides the normal termination process. In this case, the UGA codon is not truly ambiguous. Its meaning is strictly conditional and highly controlled by the surrounding architectural context. The cell is essentially leaving a giant, flashing neon sign next to the stop sign that reads, "Ignore this stop sign if you are building this specific protein." It is a neat trick, but the plot thickens drastically with the 22nd amino acid pyrrolysine. Discovered in 2002 in methane-producing Archaea, pyrrolysine is a massive, incredibly complex amino acid that these microbes use to construct enzymes crucial for digesting specific chemical compounds, like methylamines. Pyrrolysine is encoded by the UAG, or amber, stop codon. However, unlike the highly regulated selenocysteine system, researchers could not find any equivalent to the neon sign for pyrrolysine. There is no structural hairpin loop, no specialized elongation factor dragging things into place. Instead, pyrrolysine insertion relies on a brute force competition model. The archaeal cell produces a unique pyridized knots transfer RNA that recognizes the UAG sequence. When the ribosome encounters UAG, a high-stakes game of molecular musical chairs begins. The transfer RNA tries to enter the ribosome to add the amino acid, while simultaneously, the archaeal release factor tries to enter the ribosome to terminate the translation and cut the protein. For years, scientists assumed that the Archaea had simply optimized this process. They thought the microbe perhaps mutated the context around the UAG codon, or fine-tuned the levels of release factors to ensure that pyrrolysine was inserted perfectly every time it was needed, and termination happened perfectly everywhere else. But cutting-edge research has revealed a reality that is far messier and far more brilliant. In late 2025 and early 2026, researchers from the University of California, Berkeley, led by molecular and cell biologist Dipti Nayak, published a series of groundbreaking studies focusing on Methanosarcina acetiverans, a model methane-producing archaeon. What they found effectively overturned a 60-year-old biological doctrine. They discovered that this specific microbe maintains a truly ambiguous genetic code. For this tiny organism, the UAG codon has a dual meaning. It acts as both a stop signal and a green light for pyrrolysine, simultaneously and seemingly at random. When the ribosome of this archaeon reaches a UAG sequence, it basically flips a genetic coin. Sometimes the release factor wins the competition, the protein is truncated, and the ribosome falls apart. Other times, the transfer RNA wins, pyrrolysine is inserted, and the ribosome continues chugging along, building an extended protein. This creates a chaotic scenario where a single gene carrying a UAG codon produces two entirely different pools of proteins, an extended full-length version containing pyrrolysine and a shorter truncated garbage version. Under the rigid dogma of classical biology, this is catastrophic. Objectively, ambiguity in the genetic code should be deleterious. Producing truncated proteins wastes immense amounts of cellular energy. If these half-finished proteins misfold, they can clump together, form aggregates, and literally kill the cell. Yet, Methanosarcina acetiverans does not just survive with this loose translation style, it absolutely thrives. The microbe possesses between 200 and 300 genes containing the UAG codon. The ambiguity is not a failure of the system, it is a highly evolved feature. As the Berkeley researchers noted, biological systems are fundamentally more flexible than the standard models give them credit for, and in the case of this microbe, that ambiguity is an intentional survival mechanism. But why would an organism intentionally play Russian roulette with its own proteins? The answer lies in the harsh, fluctuating environments these archaea call home, and a biological strategy known as bet hedging. This microbe survives by consuming methylamines, which are chemical compounds that are abundant in decomposing organic matter, deep-sea vents, and even the human gut. To digest these methylamines and convert them into methane gas for energy, the archaeon requires specialized metabolic enzymes. Crucially, the active sites of these enzymes absolutely require that massive 22nd amino acid, pyrrolysine, to function. If the microbe lived in an environment where methylamines were constantly available all the time, it might make sense to permanently reassign the UAG codon to pyrrolysine and rely on the other two stop codons for termination. But methylamines are an intermittent food source. Synthesizing the massive, complex pyrrolysine molecule from scratch is biochemically very expensive. It takes a lot of energy. When the food sources dry up, continuing to force the insertion of expensive pyrrolysine at 300 different UAG sites across the entire genome would be an enormous, fatal waste of resources. The ambiguity of the genetic code offers an elegant, energy-saving solution. The meaning of the UAG codon in these archaea is dynamically biased by the environmental conditions and the availability of pyrrolysine inside the cell. When the archaeon detects an abundance of food, it ramps up the production of pyrrolysine. The cell becomes totally flooded with the amino acid and its corresponding transfer, RNA. By pure mass action, the sheer overwhelming volume of the transfer RNA outcompetes the release factors at the ribosome. The genetic coin becomes weighted. UAG is interpreted primarily as a go signal, allowing the cell to rapidly mass-produce the full-length enzymes needed to feast on the methylamines. Conversely, when the food disappears, the cell halts pyrrolysine production. As those levels plummet, the release factors win the competition by default simply because there is nothing to compete with. The UAG codon reverts to acting primarily as a stop signal. The cell immediately ceases the production of the unnecessary digestive enzymes, resulting in truncated proteins that are quickly degraded and recycled for spare parts. This stochastic probability-based decoding allows the archaeon to adapt to fluctuating environments instantly, without needing complex, dedicated genetic regulatory circuits for every single gene. It navigates the ambiguous genetic code in direct response to environmental cues, tuning the output of its protein factories simply by adjusting the cellular demand for one rare amino acid. To truly grasp how profound this ambiguity is, we have to zoom in even closer, to the atomic level of the ribosome, the microscopic machine where this conflict actually plays out. Translation termination in archaea is carried out by a protein called archaeal release factor 1. Unlike bacteria, which use two different release factors to read different stop codons, archaea and complex eukaryotes like humans have streamlined the process, using a single omnipotent release factor capable of recognizing all three stop codons. Structural biology has revealed that this release factor is a marvel of evolutionary mimicry. When it enters the ribosome, its physical shape closely mimics the size and the L shape of a transfer RNA molecule. Just as a transfer RNA has an anticodon loop that reads the genetic sequence, the release factor contains specific structural motifs, notably the NIKS and Y times C times F amino acid sequences that physically interact with the stop codon on the messenger RNA. The domain of the release factor protein is incredibly flexible, acting almost exactly like the arm of a transfer RNA, allowing it to adapt its physical conformation to perfectly bind to the stop signals. Once it recognizes a stop codon, it positions another vital region, known as the GGQ motif, precisely into the catalytic center of the ribosome. This GGQ motif triggers a water molecule to attack the chemical bond holding the newly made protein to the ribosome, liberating the protein into the cell. In a standard cell, when the UAG sequence appears, the release factor slips into the ribosome unchallenged. But in our rebel microbe, the release factor is under siege. It must compete directly against the pyrrolysine transfer RNA for the exact same physical space inside the ribosome. There is no manager protein deciding who wins. There is no intelligent design guiding the process. It is a totally blind race, determined by thermodynamics, concentration gradients, and the physical kinetics of molecular collisions. If the transfer RNA binds first, the ribosome undergoes a structural shift, locking it in place, catalyzing the chemical bond, and moving to the next codon before the release factor even realizes what happened. If the release factor binds first, the GGQ motif strikes, the protein is severed, and translation ends. The fact that life can sustain itself on such probabilistic molecular warfare challenges the long-held notion of genetic determinism. Context is king here, and the broadening scope of genetic ambiguity is staggering. While these methane-producing archaea provide a stunning example of context-dependent ambiguity based on metabolic states, they are not the only organisms bending the rules of the genetic code. The concept of context-dependent translation termination is rapidly emerging as a hidden dynamic across multiple branches of the tree of life. Take, for instance, certain ciliates, like Condylostoma magnum, a single-celled organism. In these organisms, the breakdown of the genetic code is even more extreme. This organism utilizes all 64 codons to code for standard amino acids, meaning it has entirely erased dedicated stop codons from its genome altogether. UAA, UAG, and UGA are all translated into amino acids. How then does it ever stop making a protein? Researchers have discovered that the translation machinery in these ciliates reads the ambiguity based on the physical proximity to the end of the messenger RNA transcript. Through an astonishing process of spatial awareness, if a stop codon appears close to the very end of the genetic tape, the ribosome interprets it as a true stop. If the exact same codon appears earlier in the sequence, it is simply read as an amino acid. Furthermore, scientists have proposed the ambush hypothesis, which suggests that the placement of stop codons is a highly strategic evolutionary weapon used by organisms to maintain genetic integrity in the face of random ribosomal errors. Sometimes, the ribosome accidentally slips forward or backward by one single genetic letter, a phenomenon known as a frame shift. If the ribosome shifts out of the correct reading frame, it starts reading the wrong three-letter groups, generating totally gibberish proteins. To prevent this, genomes are peppered with off-frame stop codons. Research across archaea, bacteria, and eukaryotes reveals that immediately downstream of slippery, frame-shift-prone sequences, there is a massive spike in the density of these hidden stop codons. They act like biological landmines, lying in wait to ambush a rogue ribosome that has slipped off the tracks. They terminate the gibberish protein immediately before it drains the cell's precious energy resources. This reveals that the genetic code is heavily influenced by spatial and sequence context. A stop codon is not just a digital command. Its meaning is actively shaped by what precedes it, what follows it, the metabolic state of the cell, and the competitive environment within the ribosome itself. The global ripple effect of this is enormous, stretching from the deep sea all the way to the human gut. The revelation that archaea rewrite their stop codons is not merely an esoteric biochemical curiosity for textbook nerds. It has profound ecological and health implications for all of us. The methanogenic archaea that rely on pyrrolysine and genetic ambiguity are major players in the global carbon cycle. Methanogens are responsible for producing nearly all the biologically generated methane on Earth. Methane is a potent greenhouse gas, trapping roughly 25 times more heat per molecule than carbon dioxide over a century. The efficiency with which these archaea digest organic matter and belch out methane is directly tied to their ability to synthesize pyrrolysine and navigate that ambiguous UAG codon. Understanding the environmental triggers that bias this genetic coin flip could provide critical insights into how methane emissions might shift in response to changing global climates and warming ecosystems. Moreover, these ambiguous archaea reside right inside you. Species of methylamine-eating archaea have colonized the human digestive tract. Every time you consume foods rich in choline or L-carnitine, such as eggs, red meat, and dairy products, the bacteria in your gut break these compounds down into methylamines. If left unchecked, these methylamines are absorbed into your bloodstream and converted by your liver into a compound called TMAO, which is heavily linked to the development of atherosclerosis, heart disease, and severe strokes. Fortunately, the pyrrolysine-wielding archaea in our digestive tract feast on these methylamines, effectively neutralizing them before they can harm our cardiovascular system. The efficiency of this protective mechanism relies entirely on the Archaea's ambiguous genetic code. When we eat a high-meat diet, the sudden spike in methylamines triggers the Archaea to ramp up pyrrolysine production, bias the UAG codon toward protein synthesis, and churn out the specific enzymes that clean up our toxic digestive exhaust. In this amazing way, a bizarre quirk of genetic ambiguity in a microscopic gut resident plays a direct protective role in human heart health. Beyond ecology and human health, rewriting the textbooks has massive implications for the future of synthetic biology. For bioengineers, the realization that the genetic code can be hacked by nature is an open invitation to do the same in the lab. If Archaea can expand their genetic alphabet to include 22 amino acids using ambiguous stop codons, humans can engineer life to include dozens more. For decades, the concept of unnatural amino acid mutagenesis has been the holy grail of bioengineering. Scientists want to build synthetic proteins that incorporate artificial chemical groups. Things like glowing fluorescent probes, photoreactive crosslinkers, or ultra-toxic payloads for targeted cancer drugs. Because the standard genetic code has no room for new amino acids, bioengineers frequently borrow the pyrrolysine system straight from these methanogenic archaea. By taking the archaeal transfer RNA and the specific enzyme that attaches pyrrolysine to it, and inserting them into standard lab bacteria or mammalian cells, scientists can artificially reprogram the UAG stop codon in these lab organisms. They essentially export the archaeal genetic ambiguity into synthetic systems. However, the insights gained from methanosarcina acetivirans highlight a significant hurdle in this process. When we force UAG to encode unnatural amino acids in synthetic organisms, we introduce the exact same ambiguity and competition with release factors that the archaea face. The host cells end up producing a messy pool of full-length synthetic proteins mixed with truncated garbage, severely lowering the yield of the life-saving drugs we are trying to manufacture. Armed with the new knowledge of how archaea manage this ambiguity natively, by balancing transfer RNA concentrations and leveraging context-dependent RNA sequences, synthetic biologists are now engineering highly optimized genomes. Some advanced labs are taking the radical step of entirely removing the UAG stop codon and the corresponding release factor from bacterial genomes. This creates what are called genomically recoded organisms, which possess a completely blank, unambiguous codon, ready to be assigned permanently to synthetic chemistry without any competition. These firewalled synthetic organisms, inspired directly by the extreme adaptability of archaea, are paving the way for a brand new industrial revolution in biotechnology. We are looking at a future where we can develop ultra-targeted antibody drug conjugates for advanced cancer therapy, or engineer highly virus-resistant agricultural crops that rely on synthetic amino acids viruses cannot process. The symphony of imperfection is what makes life so resilient. The universal genetic code was once viewed as a flawless, crystallized, mathematical absolute. It was taught as the biological equivalent of computer machine code, rigid, completely deterministic, and universally true across all domains of life. But the discovery of genetic ambiguity in archaea, particularly the stochastic, loose translation of the amber stop codon in methanogens, has permanently shattered this illusion. Life, we are learning, is not a perfect machine, it is an opportunistic survivor. The archaea have shown us that ambiguity, which is almost always viewed as the enemy of complex systems, can actually be harnessed as a brilliant evolutionary strategy. By allowing a single stop codon to mean two completely different things simultaneously, this tiny microbe achieved an elegant, highly energy-efficient bet hedging mechanism. It allows the organism to rapidly adapt to a chaotic, shifting environment without needing to rewrite its entire genetic operating system. It forces us to reconsider the genome not as a strict architectural blueprint, but as a fluid, context-dependent script. It is a biological script where the punctuation marks are sometimes completely ignored, where the end of a sentence might actually be the middle of a brand new thought, and where the microscopic translation machinery relies on probabilities, molecular collisions, and quiet environmental whispers to decide exactly what to build next. As we continue to explore the microbial twilight zone, sending submersibles down to sequence the DNA of uncultured archaea from the deepest ocean trenches and the hottest thermal vents, we will almost certainly discover even more alternative genetic codes. We may find microbes that have incorporated a 23rd or 24th amino acid, or organisms that have evolved entirely different mechanisms of resolving molecular ambiguity that we cannot even imagine yet. The story of how Archaea rewrite universal DNA stop codons is a profound reminder of the boundless creativity of evolution. It teaches us that in the messy, hyper-competitive, and ever-changing theater of biological life on Earth, being perfectly precise is sometimes a massive disadvantage. Sometimes, basic survival requires a little bit of flexibility, a high tolerance for making errors, and the audacious ability to look at a universal biological stop sign, flip a coin, and decide to keep right on going. Thank you for listening to this deep dive on Fun Facts Online, where we explore the weirdest and most wonderful realities of the scientific world.